Welcome to Click for Biology. We're looking at plant nutrition and in particular at plant experimental techniques. In this particular video, we're going to be looking at a technique which is used to measure the rate of photosynthesis. If you'd like a copy of this presentation, that's as a PDF and is linked into the description of the video, which you can download below. Let's take a look at the apparatus. Now, in this particular application of the apparatus, I'm going to look at the effect of light intensity on the rate of photosynthesis, although this basic technique can be modified by changing the independent variable from light intensity to things like temperature and various other factors. The source of light provided is a bench lamp, and we're going to measure the distance between the bench lamp and our photosynthesis apparatus. We have a boiling tube with our aquatic plant inside, and we know that with the light, the plant is going to produce oxygen. The oxygen will be given off and will pass upwards. The boiling tube is sealed here, and the gas oxygen will accumulate, passing along this catheter to the gas syringe. The gas syringe will then be pushed out. If the marker begins here, it will be pushed out a certain distance, and then we can measure here the volume of oxygen which is produced. By using a stopwatch, we can therefore calculate the volume of oxygen produced over time, which will give us the rate of photosynthesis. Whilst distance between the bench lamp and the beaker sounds like a way to measure the light intensity, this is in fact an error, and it's necessary to also use a light meter, which we place at this position here to measure the light intensity at any given distance. What we would actually find is that if we double the distance, then we square the distance would give us a quarter, and the light intensity would actually decline by a quarter if we doubled the distance between here and the beaker. This is called the inverse square law. So the way to get around this problem is to take your light meter and place it here, and then change the distance of the lamp and use the reading on the light meter, which of course would be in the units of lux, as your independent variable. This technique is often replicated in exams. Strongly suggest that you learn the setup. When you've done that, there's a 3-minute quiz here. If we pause the video, answer these questions nice and quickly, and then check out the answers in the descriptions below before moving on to the next section of the course, which is linked in here to a playlist and of course a link to the channel so that you can subscribe. And if you have a moment and you found this useful, please give us a thumbs up. Much appreciated.